Before I begin, I'm at a medical conference, so I have to give away my conflicts of interest disclosure. I've started a couple companies, advised a bunch of companies. You shouldn't believe a single thing I say after this slide. And also, a very important slide. I've been traveling in Israel. I was in Haifa before this, which means I did not get to see the season premiere of Game of Thrones, so I will kill anyone who tells me what happened on Sunday. So, a very important slide up front. So, two major questions really drive almost all of the research I'm working on in my group. And they're really basic, fundamental questions that I will argue and I think convince you over the course of this talk that we have very, very poor answers to these important foundational questions. And actually, experiencing health and disease as a patient myself really has guided a lot of the research that I do. So I have Crohn's disease, inflammatory bowel disease. This is a recent visit to, this is actually Greenwich Hospital, not Mount Sinai Hospital. It's an inflammatory disease of the bowels, and you get inflammation. You have to go to the emergency room. And I did not put this picture up to get sympathy from everybody here. First of all, getting sympathy from Israelis is a bad idea anyway. But I didn't put this up to get sympathy because Joel in that picture right there has so much morphine in his system. He feels amazing, trust me. I'd much rather be that guy right now than the jet-lagged Joel on the stage right now. The reason I'm telling you this, one thing that happens to you when you get Crohn's disease, which again is a disease of the intestines, is you get cutaneous manifestations of the disease. You get skin rashes and things like that that can erupt before you have a flare of your disease that sends you to the hospital. So I'll go to my GI doctor, who's one of the best in the world at Mount Sinai, and I say, isn't this interesting? Before my gut is having a reaction that's sending me to the hospital, my skin, I particularly get a rash all over my chest and my arms. I said, what do you think about this? And he says, I think you should see the dermatologist. Okay, so I go to the dermatologist across campus, and I say, isn't this interesting? My skin seems to be connected to my gut somehow. What do we think about this? If you think about it, your skin is sort of, or your gut's sort of your inside skin. I know that's kind of gross to think about, but it is. So I asked the dermatologist, what do you think about this connection? And he says, I specifically became a dermatologist to never think about the colon, so please take a steroids and get out of my office, right? So as a patient, I experienced, even though this disease was happening on me, I experienced being bounced around silos, and this is really how we think about disease even today, right? A symptoms and anatomy-oriented view of disease. One of the papers I did actually back when I was a graduate student was shown here, the figure shown here, I'll explain in a second, was to take all of the emerging molecular data that was being generated, gene sequencing data, gene expression data, and ask the simple question, does the molecular taxonomy of disease look anything like the symptoms and anatomy-based taxonomy of disease that we still organize our health system and our research around? And what you're seeing here is if two diseases are connected by a line, like Alzheimer's disease and actinic keratosis here, which is a skin disease, it means that at the genome-wide level, in terms of the pathways and things that are being disrupted, at a genome-wide scale, they share more in common than you'd expect by random chance. So it's very strange that Alzheimer's disease and actinic keratosis would look similar to each other at the molecular level. And what we find, again, malaria looking like Crohn's disease. So the molecular taxonomy of disease suggests that the way we think about disease at the gross anatomy level, they're not similar to one another. This is perhaps, that's an incorrect way to think about it. And we've done a lot of work using these types of things for drug discovery and things, I won't mention it. But you can take this concept of using data-driven approaches to map out how we think about disease in a different way up at the patient level. So we have a very nice resource at Mount Sinai called the Mount Sinai BioMe Biobank, run by Judy Cho and the Institute for Personalized Medicine. So for about 50,000 of our patients now, which are sampled from our outpatient population, they're consented to give DNA. And of course, we have all their electronic health records. We have genome-wide sequencing information available on them. And they're consented for recontact, which means that we can actually call them back in the future and ask for additional information, such as perhaps microbiome or RNA to be collected. But right now, we have lots of data on EHR data and DNA. So one of the first projects I worked on when I went to Mount Sinai, I was very interested in the concept of, which is really a buzzword now, of sort of a learning healthcare system, right? So if I go to Mount Sinai, let's say, or I'll keep picking on my own institution, even though it's great, I won't get in trouble with anybody else. So, they'll say, if I go to the doctor, they'll say you're a 42-year-old Caucasian male, family history of this, disease history of this, complaining about this, let's treat you based on perhaps guidelines. And I'll say, wait a second, you see several million patients every year. Which patient looked like me and what happened to that patient? Sort of like when you go shopping on Amazon, they'll say, okay, there's not some expert sitting back there picking the products, right? It's being based on my browser history, being based on the other sites I visited, shoppers who look like me. So what you're looking at here is not a network of diseases. This is actually a network of patients. And the X and Y doesn't mean anything here. The topology is the only thing that's important. So what we did is we took, for all these patients, which is again an outpatient population, took all of their quantitative measures in the electronic health record. So height, weight, CBCs, metabolic panels, anything that was a hardcore, or a quantitative measurement. I ignored things like diagnosis codes and things in this study because I considered those to be subjective in some way. So then I represented each patient by a few hundred of these quantitative variables that we measured, and I basically said which patient is similar to this patient, similar to this patient, on everything we ever measured on them. And this is a patient-patient similarity map. Of course, it's a 100-some dimensional, or 200-some dimensional space crushed down to 2 dimensions. We used a method called algebraic topology, which I don't have time to talk about today. But the point is, this is a patient-patient similarity map organized by the similarity between patients based on their data. And the idea is, since I'm in Israel, I'll say, where is your little Waze dot blinking if you're a new patient? And does it matter if you're here or over here? I can tell you don't want to be over here. This is where the sickest patients tend to be. But now the coloring here is important. Then after the fact, after we organized patients based purely on their data, and in this unsupervised way, we said, where are the patients with type 2 diabetes in the health records? By the way, that's a very hard thing to do, figure out who has type 2 diabetes, by the way. Computer scientists often think, oh, we'll just query the health records and find the patients that have type 2 diabetes. It's a very hard thing to do. You actually have to build a very complex rule because the ICD codes, for example, are biased in many ways. But we have lots of type 2 diabetes patients. We simply asked, where are the patients with type 2 diabetes? This is a disease we think of generally as one disease. And then we get these hot spots. The red here means type 2 diabetes patients are found in this region more than you expect by random chance. If you just focus on the type 2 diabetes patients alone, the provocative idea behind this particular work was, did we have type 2, type 3, and type 4 sitting under our nose the whole time, but we didn't know it because we didn't look at our patients in a data-driven way. So you can cluster data all day long, and you get cool figures, but who cares if it doesn't mean anything clinically, right? So we have electronic health record data on these patients. We can ask a question like, if a type 2 diabetic was found in this group, do we have any increased or decreased risks with comorbidities, for example? So what we showed is that if you're in this group, you had increased risk of stroke and heart attack above and beyond the typical type 2 diabetic. If you're in this group, you had increased risk of cancer. All diabetics have increased risk of cancer, but if you're in this group, you had risk above and beyond the typical type 2 diabetic. And in this group, there was increased risk for whatever reason we don't understand of allergy and infection. I remember we had genetics on these patients. The genetics was not used to inform the clustering of this. Later on, what we did in the study is we actually found genetic markers that uniquely identified each one of these groups. And for example, if you looked at the genetic markers that were unique to this group, you actually found that they were enriched in things like vascular endothelial signaling pathways and things like that, suggesting that it wasn't just an artifact of the data analysis, retrospective data analysis, that there was potentially some genetic markers. So a big limitation of this study was that it was cross-sectional in time. We actually have a new version of this method that we're about to publish that looks over time. It's almost like looking down this in 3 dimensions. So, but we have lots of really cool data we can use in the health system now, right? We have electronic health records and billing systems and lab ordering systems assembling a huge amount of data. We have now things like genetics. So the big question, of course, is how are we going to use this information to build new types of predictive models in a scalable way? So I'm sure there's been a lot of talks about deep learning. There's a lot of booths about deep learning. But I always like to ask the audience who's heard of deep learning, and of course everybody's heard of it today. But people are afraid to admit they don't actually understand what it means. So I always like to use this Google cat video example, right? So when deep learning, which is obviously a form of machine learning that's enabling some of the new types of capabilities we have in medical imaging and machine learning, came out, there was this article in the BBC, Google can make an algorithm to find cats in videos. And of course, my mom emails me and says, who cares? Like, what is the point of this? Why is this in BBC? So let me explain to you why this is a big deal. So imagine if you wanted to use the previous sort of generation of machine learning to build an algorithm that would find cats in videos, what would you do? Well, you'd have to find a cat expert, which is, like my daughter, for example, and you'd have to get millions of videos of where there's cats, and frame by frame, first of all, you'd have to decide what features are important for a cat. The ears, the eyes, the paws, and you'd have to get a bunch of examples, and you'd have to hand annotate frame by frame a huge number of examples and feed that human-trained data set into an algorithm and train it, and hope that you made an algorithm that could find cats in videos based on the features you chose and the data you put in. But that's not what they did in this case at all. They took millions of cat videos, millions of videos that had cats, millions of videos that didn't, and they told the algorithm, you figure out what a cat looks like. They did unsupervised feature learning and was able to just learn purely from the data what a cat looks like. And of course, if you're interested in many of the types of products that are out here, and certainly a lot of the research that was discussed here today, Eric Topol put out a very nice book recently called Deep Medicine, which is sort of a popular science book to explain how deep learning is being used in medicine. It just came out. And we're lucky enough to be featured in this book. One of our papers is featured in this book, a deep patient. And the question here was, this is actually very similar to the Google cat video. We asked the question, could we do unsupervised deep feature learning on electronic health record data? So say, for example, you wanted to build a model that predicted who's going to get new diagnosis of diabetes in the next 30, 60, 90 days. Well, one thing you could do is talk to a diabetes expert, and they would tell you, let's put HbA1cs in, let's put in fasting glucose, let's put in these drugs, and let's try to see if that model will predict who's going to get diabetes. The other thing you can do is what we did here, which is let's take everything in the electronic health records and just throw it into a deep learning framework and have it learn characteristics of our patient population in an unsupervised way. And then what we did is the task was, can we predict who's going to get new diagnosis of some disease in the next 30, 60, 90, 180 days? The point of this paper was we were able to build fairly well-performing models that predicted who's going to get new diagnosis without, of course, talking to a single expert, right? So we did unsupervised feature learning on the HR data, and then you could build these supervised predictive models. So there's another reason we need to start thinking about, not only for building predictive models, but also for biomedical discovery using more of these sort of unsupervised or somewhat unbiased data-driven approaches. It's to save us, which I think is a huge problem in biomedical research, from the streetlight effect, which is sort of explained by this cartoon here, where this person's looking for the quarter that they dropped under the light. The cop asks, why are you looking here? Because the guy says he dropped it 2 blocks away, and it's because the light is shining better here, right? So if that doesn't resonate you, the same idea is presented here by our former Secretary of Defense, Donald Rumsfeld, which, with this quote he got made fun of for, but it's actually a pretty good quote to explain this concept that I'm talking about, is the known knowns, the known unknowns, and the unknown unknowns, right? So a huge problem, in my opinion, in biomedical discovery is that we tend to iterate from things that we know, but we're very bad at sort of managing for these unknown unknowns, which are more discoverable now because we have huge amounts of data. So one example of this is a study we published recently on Alzheimer's research. So I'm showing you this not to show you that we got a quote, or our paper was covered in the New York Times, but I got the most ridiculous quote posted in the New York Times, which is, I went looking for drugs, and all I found were these stupid viruses. This is a quote. So I'm going to explain what this means here in a second. So for those of you who don't know about Alzheimer's, there's a lot of, we don't really understand the disease very well yet. There's a lot of things that contribute to it, like oxidative stress, et cetera, but a lot of the biology or discovery is focused a lot on tau pathology, in particular the formation of these characteristic amyloid plaques that we find in the brains of Alzheimer's patients. Even if you personally don't have risk for Alzheimer's, or your family doesn't, you should all care about it, because if any disease is going to bankrupt our country, it might be this one. And of course, we can even image these amyloid plaques in the brain. And a lot of the drug discovery is then focused on eliminating those plaques from the brain, the amyloid plaques, thinking we could reverse the disease. But of course, that has been a colossal failure of a hypothesis. And in fact, I think the final death throes of the amyloid hypothesis might have been the latest failure of the Biogen drug that was just pulled. So one suspicion that I had, and many have, is that the plaques are just the downstream consequence of some upstream mechanism we don't fully understand. So we had a great opportunity last year where the National Institutes of Health in the US engaged in this project called AMP-AD, Accelerating Medicine's Partnership for Alzheimer's Disease, where they really got a bunch of brain banks together and measured almost everything they could imagine to measure on these Alzheimer's patients. It was pretty amazing. And then we were brought in after the fact to analyze the data, given what we focus on. So we analyzed it in a different way. We didn't just say, let's look at this dimension, let's look at it from the genetics perspective, the microbiome, whatever. We used network modeling. So instead of, again, looking at one dimension at a time, we'll say, imagine this is a DNA variant that's changing in the population of brains that were measured. And maybe the yellow is a metabolite or a protein. And as we see the elements in the data sort of varying across the data set, we'll just draw a correlation sort of between them. But there's actually methods that we have later on to turn these correlation networks into sort of directed networks. So again, instead of just building a model of the DNA, a model of the protein, et cetera, we build a holistic model that incorporates DNA, protein, RNA, imaging, et cetera, to sort of, and learn that purely from the data, not based on what we think. We don't look at textbooks and those pathways that have been published on Alzheimer's. We see, what does the data tell us about how Alzheimer's is put together across these dimensions? And then we asked a very simple question, which is taking these multi-scale networks of healthy brains, which again represent DNA, protein, et cetera. I mean, it looks like this. You can't see anything. It's a big hairball. But then also the same network learned from the Alzheimer's brain data. And then we asked our algorithm to say, tell us what's most different between this network of information and this network of information in Alzheimer's. And one of the top things that came back was something that looks like this, which is HHV6, roseola virus, part of the herpes virus family, basically had learned a network, was telling us that viral genes were interacting with host genes and potentially contributing to the pathogenesis of Alzheimer's disease. Something we totally didn't expect. What we were actually looking for was new drug targets to treat Alzheimer's disease. The last thing I wanted to find was that viruses might be contributing to Alzheimer's disease, because it got me in a huge amount of trouble with all the Alzheimer's disease researchers who hated everything about this paper because it went against the grain of what's known. We found a bunch of new pathways. We ran a bunch of experiments, and we actually showed that virally responsive mRNA, microRNAs and things were actually contributing to beta amyloid plaque formation. And it actually seems from our work and other people's work that really innate immunity is at the root of Alzheimer's disease. And I'm happy to say one of the great outcomes that came out of this is the NIH just introduced a new high priority RFA for the infectious etiology of Alzheimer's disease. This would have been unheard of just a year ago, but the overwhelming evidence forced their hand to start looking into this now. And in fact, one of the sad things that happened when we published this paper, there was a bunch of researchers who said they had a bunch of data that supported this hypothesis, but they're too afraid to publish it because they were afraid of retribution from the neurology community. So, okay, so we can do all, I just talked about, we can do all these kinds of cool things if we have lots of data, right? Like the deep learning, or particularly like with the Alzheimer's modeling. So what sources of data do we have to play with? So I always, people get excited, especially when they come from the outside of the health field, about electronic health record data. Oh, we can do all kinds of amazing things with this data. But let's not get too excited about this. I want you to imagine the EHR data as sort of a spreadsheet where you have patients in the rows, EHR data fields sort of in the columns, and stack those spreadsheets by time for every day of data we have. I'll give you a clue. Does anybody have an idea how empty this data cube is? Right? It's super, maybe you could be generous and say it's 95% empty, and Matt and David would have good answers for that. It's a very empty, sparse data set. The diabetes work that we did and the deep patient work, I'm shocked we got anything useful out of this data, to be honest. It's so sparse. There's so many biases built into it. So it's definitely not the solution to our problem. Another thing that we don't have a lot of in the health record is longitudinal data, right? And data on patients sampled over time in a very consistent way. Why would we want this information? Well, one of the big limitations we have in using genetics to inform discovery is that while genotyping and genomic sequencing is now totally commoditized, we're missing the dynamic phenotypic data to layer on top of the genetic data to really understand genomic function. And I don't have too much time, but I'll give you a short genetics lesson on why we need to collect this longitudinal information. So there's something in genetics called expression quantitative trait loci. Sounds super complicated, it's not. All it means is there's situations where variation in the DNA, so you see this SNP, people who have 2 copies of A at this single nucleotide polymorphism have low levels of mRNA expression of some gene, and people who have GG have high levels, and people who have the heterozygote have medium levels. Why do we look for Eqtls? Because if there's correlation between DNA and RNA expression, we think there might be a functional relationship between the DNA and the RNA. So this is one of my favorite papers to express this concept of the context dependence of genetics. So what they did in this paper is, again, looking at Eqtls. So this RS number represents one SNP in the human genome that we look at, for example, in GWAS studies. And here you see a classic Eqtl relationship. So, and this is on the y-axis is showing mRNA expression levels of this gene HIP1. It doesn't matter what the gene is. But you see that people who have 2 copies of AA genotype at this particular SNP have high levels of expression of HIP1 on average. People who have AG have medium, and people who have GG have low. Classic Eqtl relationship where we see a correlation between DNA and RNA. They stimulated these same cells from the same individuals with LPS, lipopolysaccharide, which induces an inflammatory phenotype in the cells, measured the same cells 2 hours later, the relationship's completely gone. And actually, if you look at the same cells after the same stimulation after 24 hours, it's the exact opposite direction. So someone could have been looking at these cells in steady state in a dish and published a paper that said, oh, GG genotype is associated with low levels of expression of HIP1. Well, if this was a cytochrome P450, someone might even propose we do a pharmacogenomic study based on this, but it really depends on when you're looking at it and what the stimulation is these correlations between genotype and molecular phenotype in this case are highly ephemeral, highly context dependent. They will only reveal themselves not through cross-sectional studies, but through stimulation studies. Another reason we need longitudinal data in healthcare is for challenges like this. So this is a PNAS paper that was published very recently. And we have a paper coming out showing this is also true in cardiovascular disease. But what they found in this paper is they're looking at endpoints associated with anhedonia, depression, endpoints that are used for drug discovery and depression. And what they found was that the within individual variance for these endpoints was bigger than the between individual variance of these traits, right? So the intra-individual variance bigger than the between individual variance. If you're doing cross-sectional studies, big problem, right? So we don't know what the true variance intra-individual is for a lot of these different traits. Obviously, though, we have great opportunities because digital phenotyping has become mainstream. I don't have my Oura ring on me today because I brought the wrong charger, but one of my favorites, we have the Apple Watch, even have things like the naked scale, which I happen to have in my home, which is a scale that puts you on a 360-degree scale, spins you around and builds a 3D avatar of your body. I call it the world's most unforgiving bathroom mirror, by the way. So I actually have one at home. This is a picture of me using mine at home from before I came on this trip. So, but no, the point is, obviously we have these tools, that's not me, sadly, but to digitally phenotype. Okay, maybe this will help us solve the challenge of filling in from this dynamic phenotypic information. We were lucky enough with Eric Schott and others at Mount Sinai to be one of the founding or early members of the Research Kit project with Apple, which put software in all the 1 billion iOS devices to conduct very interesting studies, like our Mount Sinai Asthma Health app. So instead of having the sparse EHR data, we can conduct the study at large scale in lots of individuals, where we get activity tracker information, Bluetooth inhaler information, spirometry data, and geospatial. We can pull in the pollen count data, the pollution data. That sounds pretty cool. And we published a paper in Nature Biotech that revealed all these cool patterns, such as forest fires triggering asthma events and things like that. But my favorite finding from this study is actually a negative result, which wasn't highlighted in the paper, which is people stop using this app pretty rapidly after they enroll in the study, right? So compliance is the biggest issue probably in healthcare, and it's no different in digital health. So great research tool, still a huge challenge in terms of managing chronic disease, right? So compliance and dropout still a huge issue, even with this big Apple Research Kit study. Another thing we're quite focused on, which I'm setting up for the institute we work on, is I'm obsessed with a graph with this type of information where we see in the US the proportion of the population with number of chronic conditions by age, so 1, 2, 3, 4, 5, 6, 7 chronic conditions, about 1.36 trillion of pharma R&D, and about $39 billion of NIH funding is spent right here. In the US, we spend about $4 per citizen on this part, on how we make that part bigger, which seems absolutely insane to me, that we don't take the same precision. He's upset about that too. We don't take the same rigor of precision health that we apply to understanding chronic disease. Not that we shouldn't focus on that, but we're so imbalanced that we don't apply it in this region. And how do we make this blue space, in this case, bigger, right? So all that was a very long setup for why I petitioned to create this Institute for Next Generation Healthcare, which is how do we begin the foundations of 2025 medicine and healthcare in practice today, not by iterating from 2019 medicine, but by trying to start over from scratch in some ways, or having the freedom to think in that way. So I would argue that most of the big data in AI and healthcare today is represented by this figure right here, that we're still very much in the stage of building a mechanical horse to pull a carriage. What do I mean by that? We have new technology, AI, machine learning, imaging, and we have a very old system that hasn't changed that much. So we're trying to shoehorn new technology to work in an old framework. So we're all patting ourselves on the back, including me, for building these compromised solutions that have new technology trying to work in an old framework. So one of the things I'm very focused on now is how do we get rid of this carriage? Right? I think we can all appreciate that the pace of healthcare service innovation is glacial at best, right? So this is a real timestamp photos of a doctor's office from 1980 and 2019. I chose 1980 arbitrarily because I was 4 years old in 1980, and I remember going to the doctor for the first time and being traumatized for some reason, maybe a vaccination or something simple. But it's pretty shocking that it hasn't changed that much. And again, I'm talking about the service level of healthcare, right? And I thought it was pretty funny the other day to see this article in the news that the NIHS basically had to pass a law to phase out fax machines from healthcare. And by the way, they gave everybody until 2020 to get rid of them. It's pretty amazing, right? So my argument is that hospitals are like old mainframe computer systems from the 1970s and 1980s. The electronic health record system or medical record systems like Epic are the operating system of this legacy mainframe. So, and now we're trying as a hack to retrofit interoperability into these things that didn't have interoperability from the beginning. So there's the FHIR protocol, which is now trying to be built in a sort of hacked in a way into retrofit these old systems that weren't built for operability, to make them interoperable. And by the way, Epic Systems, if you are not aware, is one of maybe the biggest monster out there in terms of the EHR systems. But it wasn't until Apple that the 10 trillion ton, I don't know how gorilla came along with all its might and started smacking this guy in the face, did they finally even start to move on implementing the FHIR protocol, and they have the poor patient here stuck in the middle, right? So the idea here is we're trying to be so instead of thinking about ourselves as this brick and mortar mainframe system, we should try to think of ourselves as the Intel inside of a new type of healthcare system. And what's clear to me is just like as we went from mainframes to the PC revolution, is that healthcare is rapidly moving towards decentralization. So I think many of you might be aware of the merger between Aetna and CVS, which has distribution into all the neighborhoods. They're getting their butts kicked by Amazon on the retail setting, so they're going to use their valuable real estate to deliver more and more health services. So we'll see perhaps primary care, urgent care moving out to the neighborhood with CVS, and if it's not CVS, it's Walgreens or Walmart, right? The grocery stores. These guys have the distribution into the neighborhoods, right? And one of the most interesting areas, in my opinion, for innovation, particularly around disease prevention and chronic disease management, will be in the United States with self-insured employers. Why? Is because that's where you see the great alignment of incentives. And this is probably part of the reason why Berkshire Hathaway, Amazon, and JP Morgan aligned forces, is because all the self-insured employers who are actually paying the medical bills for their employees realize that they actually have the incentives, or at least some of the strongest incentives for prevention and chronic disease management, right, in terms of financial incentives. So this is another market trend that goes into our thinking about what we're building at Mount Sinai. As David mentioned, I'm going to talk about Lab 100, which is the first one of the early tangible results of this new way of thinking. So a couple of key questions we're going to try to answer. I'll explain what Lab 100 is in a second. But imagine if we actually had a health record. So I'm joking. We always talk about electronic health records, but they're mostly records of your diseases and your treatments, right? They're actually not records of your health, right? So what if we had a health record? What would we measure? Why would we measure it? Why would it be valuable? Why would people, before they get sick, feel compelled to participate in this experience or this clinic and give information? What is the value exchange there that would motivate people? One important thing about Lab 100 is it's a real clinic that's seen patients, is open to the public for people to voluntarily participate in. You can always pay people to participate in studies and actually collect a bunch of information, but we wanted to see, could we create the value, the exchange, and the incentive that would incentivize people to voluntarily come in and participate in this system where we're trying to actually build a longitudinal health record. There's also some important design considerations before Lab 100. I always like to put this up to remind my students, who are mostly all data scientists, that stories are more powerful than data and relationships are more powerful than stories. I think everybody's very aware now, both here and in the United States, that facts aren't very motivating or compelling in any form of life. So even though we're talking about data, we need to think about how we use data to support stories and relationships, and that we sort of think of ourselves as using technology to facilitate better interactions between the individual and the care agent, and not pretend, for example, to replace the doctor. So I want to show you a video of Lab 100, which is this smart deep phenotyping clinic that we built at Mount Sinai. I showed the video because everybody thinks it's a 3D rendering when I show it, but if you come to New York, you can participate in this clinic that we built. And the reason we built Lab 100 was realizing that the problem wasn't that we needed better algorithms to analyze the data. It was realizing that, we probably have all the algorithms we need. The problem is that we're not collecting the right data to do the types of predictions that we want. So as a data science-minded person, I shifted my mindset completely to realizing, we need to actually collect the right data, which is always the case, by the way. Machine learning, so TensorFlow is fine. We need to build a deep phenotyping clinic that's collecting millions of digital data points per patient encounter, but is also incentivizing patients to participate. So this is when you sign up for Lab 100, you sign up on the iPad, and this is your initial patient consult area where you come in. And then what happens is there's various stations that measure different aspects of your health with deep digital phenotyping. This is, again, the initial patient intake area. One thing we do in Lab 100 is every time we measure something on you, we show where you fall in the population distribution of people like you, your age, gender, ethnicity, income level if possible, et cetera. So we don't just show you numbers, we show where you compare to peers who are most relevant to you, all right, in this sort of design principle. We do cognition and dexterity, One thing that's really weird is we have some really old things in the clinic, like this is an old Pegboard dexterity test, but we're able to actually to make it new again because this guy doesn't even need to be here right now because there's a HD camera above that scores the whole test. So Pegboard dexterity test is an old validated instrument, but at the same time we're getting HD video that we can use machine learning on. So the principle was measure things that are well validated that we can say something to the patient today and have lots of studies, but at the same time collect millions of other digital data points that we can later go back and mine in a more prospective way or in a research setting. 3D body scan, balance assessment, which is scored by the Xbox Kinect camera. Obviously, I don't need to tell a lot of people about the importance of falls and balance. And we even do blood testing inside Lab 100, and one of the principles behind Lab 100 is instant gratification, right? We live in an instant gratification world. Every time we measure something on you in Lab 100, the results are instantly interpreted and displayed in front of you the second we've measured it, right? And put in context, right? Because at that point, people are thinking about it and it's more relevant. Even blood testing is done on site and is done and interpreted before you're done with the visit. None of this will call you if something's wrong or out of range type of thing, right? Everything's instantaneous. It's an important design principle. So you go through these different digital phenotyping assessment levels, and then there's all the data gets synthesized into the storyboard about your health. And instead of the physician, which we think about quite deeply too, or the clinician, being a data entry clerk, you now are spending most of your time face-to-face with a patient. The clinic itself takes care of the data collection for the most part. You're left to actually have this connection with the patient. And we found all kinds of really interesting things. One, that having all this data in context and sort of telling a story about the patient's health has caused patients to ask a lot of questions they wouldn't normally ask. And also, our user satisfaction is extremely high. Our net promoter score is something like 9.6 or 7 or something like that, extremely high. And strangely enough, people 50 to 70 love this thing. I thought it would all be young people. People age 50 to 70 love it. We've run several hundred people through it already, and we have a waiting list of about 1,500 people long right now. But if you're going to build a clinic of the future, you shouldn't just build this one, or at least the health assessment clinic of the future. You can't be satisfied only with a clinic, so we're thinking more about the ecosystem around this. And one of the things we believe is that you need to be able to, of course, if you assess the health risk, you need to be able to capture outcomes of any interventions. One of the things we do a terrible job of, because of the way the system is set up in the US health system, is to, of course, capture, we often don't know if someone filled their prescription, much less took the drug, et cetera. So how can you use things like reinforcement learning and things like that, fancy machine learning, if you don't have a closed loop system? So we envision that every intervention from Lab 100 would be deployed as an N of 1 clinical study. N of 1 clinical study means a clinical study on a single individual, right? And we actually just published a paper, Beth Percha, who's in my group at Mount Sinai, on sort of the statistics behind conducting a single person clinical study. But we actually spent 2 years building an app called the N of 1 mobile app that's in clinical trials right now that would let someone like my mom, who's technically unsavvy, actually receive an intervention as an N of 1 study. And actually, she doesn't feel like she's conducting a study on herself. So it's a very easy to use system for conducting N of 1 studies. And in the background, there's a statistics engine that takes care of all the complex math, and then the end creates a very nice user interface for conducting an N of 1 study and helping people interpret the results. But the point is, we want all these interventions to be deployed as N of 1s, and we aggregate the experience of all these N of 1 interventional studies that are just captured as part of the normal participation in Lab 100. So one division of Lab 100 of one clinic at Mount Sinai is a super, super boring vision, actually. So when you can come see Lab 100, it looks very permanent and very nice, like an Apple store. But then I'll tell you that we actually built it in 2 weeks. Lab 100 is not a clinic at Mount Sinai. Lab 100 is a platform for building smart clinics of various shapes and sizes. This is the whole idea behind it. It has to be scalable from the beginning. I could build one here in 1.5 weeks, no problem. So the second version of Lab 100 that we're building looks like this, like a big shipping container that could be dropped off in an employer's office, for example, right? Third version of Lab 100 we're working on that looks something like this kiosk or like this. And if you don't believe me, this is actually the design that we're building soon. Actually, Xbox has Microsoft has a new Kinect camera that can run almost all the tests we run inside the current Lab 100 from this Azure connected camera. So we have Lab 100, which is the in-person assessment, but obviously the other part of healthcare being decentralized is the sort of direct-to-consumer version, such as telemedicine, right? This is one of my favorite companies, by the way, 98.6, that is making really smart use of chatbots in the telemedicine. Telemedicine's not new. There's a bunch of telemedicine companies, but I happen to like the way that these folks employed chatbots in a very smart way. Another interesting thing that's happening in the United States, though, that's relevant to this more consumer-oriented trend, is there's a company called Hims, which maybe you're not aware of if you're not in the US, which is doing direct-to-consumer prescription medications, in this case for skincare and hair care and things like that, right? So these are medications that require a prescription. They're generic, like finasteride and things like that, but now they're marketing direct-to-consumer, and there's a physician network in the background prescribing these drugs. Another company, has anyone heard of Get Roman? I like to try to catch people on that one. No one would ever want to admit that they know what this is. Why? This is a direct-to-consumer erectile dysfunction drug company. So sometimes I can get people to raise their hand and take it down real quick, right away. So what Get Roman is doing is now taking prescription erectile dysfunction medications and marketing direct-to-consumer, right? Not even going through the health system. So it's not one thing to have telemedicine, but now you can actually do a prescription drugs direct-to-consumer by having physician network in the background and a digital platform for dealing with side effects and things like that. So I actually have my hat in the ring on this, by the way, so you shouldn't listen to me because this is a conflict of interest. So I've started a company myself in this space, which is going to be doing, we already have a direct-to-consumer microbiome test on the market. We have a blood draw device that's at the FDA right now, licensed from GE HealthCare, which will allow direct-to-consumer blood draws, and also a whole genome in partnership with a company called GenCove in New York for $49 that we'll be rolling out. So we're doing now engaging direct-to-consumer with microbiome, blood, and genetics. Again, so really the goal of what I'm trying to accomplish personally is not to try to convince these existing healthcare systems to change and move these big ships. It's to create a parallel internet of healthcare that competes with the existing system, is what I'm trying to do, which is much more directly engaging the consumer. I think we can scale to much bigger data much faster by combining things like Lab 100 with these direct-to-consumer services. So, and by the way, I think this internet of healthcare, all the pieces are here, by the way. So it's not going to be that we're going to have these EHRs and then we're going to connect the EHRs and all these cool things are going to happen. I don't think that's the opportunity. I think that's boring, actually. I think what's more interesting is a company like Clear. Who knows what Clear is? So Clear is this interesting company in the United States which gets you through airport security faster, a biometric company, right? So you get your fingerprint and your eye print. They have Department of Homeland Security, highest levels of certification, but they're rapidly expanding out from airport security. In fact, now Hertz Rent-A-Car lets you use your Clear fingerprints to just pick up your car in the lot and drive away. They're actually in baseball stadiums. Now you can buy beer in the United States with just your 2 fingers because they know they can verify who you are biometrically and they have your credit card on file, right? So I've been talking to Clear. I think the more interesting opportunity is going to come from something like this. So Clear has your biometric identification and it's going to start to have these network effects of all these different things that's going to start to authenticate you. So if you go to your Lab 100 kiosk, you authenticate with Clear. All your health record information is stored in this secure system, which has, also can be connected to your purchasing information, right? And it's with this company that actually can't mine the data in the way, for example, that like Google would, the way they're set up. And maybe it's connected to your Minute Clinic. And maybe when you go to Mount Sinai, instead of filling out the 30 forms that you fill out every time you go there, and they pretend like they've never met you, this is true of every health system, that you just put your 2 fingers on the Clear thing, and it's automatically connected to your insurance information. They can verify your insurance eligibility, et cetera. So actually, we're actually talking to Clear, which is based in New Jersey, to see, is this really the health record of the future, where we have your airline information, your credit card information, your insurance information, et cetera, connected to this system, Clear, whose whole business model, by the way, is keeping this all secure and not selling it to anybody. So this is what I'm focused on. Thanks for listening. Yeah.